Hi, Miss Nikki here. We're going to cover part 2 of chapter 4. So we're going to talk about cell communication, and we're going to talk about cell structures. So inside the cell, what different structures exist and what's their function. And then we're going to talk about cell division. And lastly, we're going to talk about, well, kind of an extension of this, we're going to talk about cell cycle. Here's an image from your textbook for the structure of a cell. We used it to talk about plasma membrane and nucleus and cytosol. So remember, all this jelly-like substance is cytosol, and inside of it are all of these cellular structures. So nucleus, DNA, nucleolus, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus. These jelly beans are the mitochondria. We're going to talk about lysosomes. So I'm going to go through the next couple of slides and we'll talk about the function of all of these structures, and then I'll put this slide up again and we'll look at them again. So we like to break organelles into membrane-bound or non-membrane-bound. So these organelles are compartments within the cell. If they're surrounded by a membrane, then they can isolate their environment. So we could have a cell, and inside the cell we have an organelle, and that organelle, we could have reactions taking place, and if we need to isolate those reactions, we're going to have a membrane around it. You're going to see that there are other structures inside the cell that are not wrapped in a membrane. Well, we're talking about membrane-bound organelles. The first one that we're going to talk about is endoplasmic reticulum. So it's kind of an interconnected network, really. It varies in shape. It extends from the nuclear envelope to the plasma membrane. You're going to see ribosomes attached in some cases. So if you see rough ER, that's endoplasmic reticulum with ribosomes on it. If you see smooth ER, it has no ribosomes. We have to talk about two different types of ER because they have different functions. So the rough ER has ribosomes that are involved with protein production. So remember, we have DNA and it gets copied into RNA, and then that RNA gets processed into an amino acid code that eventually becomes a protein. So part of this process from RNA to protein, you need ribosomes to do the job. So this is kind of the site of protein production. Smooth ER is going to have more to do with lipids. It does have other functions. We'll talk about developing carbohydrates from non-glucose sources, but we won't talk about that till we get to 202. So it's called gluconeogenesis, but smooth ER can be involved in that. Really, for smooth ER, I would think about the creation of lipids or transporting lipids or the storage of lipids. So when you see smooth ER, think lipids, and when you see rough ER, think protein production. The Golgi apparatus is where the protein ends up after the rough ER. So you had rough ER, and then the protein is going to go from the rough ER to the Golgi apparatus. So this is, again, a sac-like membranous structure. It's going to modify, pack, and sort the proteins and then secrete vesicles that contain those proteins, and they're going to go somewhere else in the cell. So some people like to think of the Golgi apparatus as kind of like the post office. It receives this protein, it's going to package it or sort it for delivery. Lysosomes are membranous sacs that contain digestive enzymes that were formed by the Golgi apparatus. So basically it's this little vesicle, and inside of it, it has digestive enzymes. So it'll participate in digestion of unneeded substances. So kind of think of it as the garbage disposal of the cell. If you have a damaged organelle, you need to dispose of it. You don't want it taking up space in the cell. So the lysosome can engulf that item, and it can use the digestive enzymes to break it down into smaller pieces, and then those can be transported out of the cell as waste. Peroxisomes are really interesting structures. They're enclosed sacs, they're smaller than lysosomes, and they come from the rough ER. So just like lysosomes came from the Golgi apparatus, these are pinched off vesicles from the rough ER. And they have lots of metabolic functions. They're kind of interesting because they do chemical digestion. So the reason they were named is they help take peroxide and break it down into water and oxygen. They're also used in lipid breakdown. They have here beta oxidation, but basically breaking down fatty acid tails. And then interesting enough, they also help with lipid synthesis. So remember we said smooth ER does lipid synthesis. Well, peroxisomes can also do some lipid synthesis. Finally, we move on to the powerhouse, the mitochondria. So this jelly bean shaped structure, what's interesting is this one has a double membrane. So it has an outer membrane and then it has an inner membrane. And this is the main source of ATP generation if you have oxygen present, right? So aerobic cellular respiration. Remember, all the electrons got stripped during the first 3 phases of cellular respiration, and then all those electrons get dumped into the mitochondria, and it generates the most ATP molecules at this end stage, the electron transport chain, right? So these are the powerhouses of the cell. And something to notice, if you see a cell that has many mitochondria, then that's probably a cell that needs lots of ATP. So here's just a normal cell. Let's say we had a skeletal. Oh, I can't do, there we go. It wasn't working there for a second. So we have this long cylindrical skeletal muscle. We're going to have fibers running through here, protein fibers. There's mitochondria shoved in every nook and cranny. So you're going to see tons of mitochondria in a skeletal muscle. So if you see 3 versus seeing 20, that should tell you that that cell is requiring lots of ATP production. Before we move on, let's look at the structures one more time. So we talked about the rough endoplasmic reticulum, and we said that this is the site of kind of protein assembly. Where did it get the instructions from? Right? DNA is inside the nucleus. DNA copies itself into RNA, and that RNA comes out the nuclear pore and goes into the rough endoplasmic reticulum, and we start assembling the amino acid or proteins. Then what happens to the protein? The protein gets deposited at the Golgi apparatus, this green structure here. It gets modified and packaged and folded, and then we ship the protein somewhere out in the cell or outside of the cell, either inside or outside, via this vesicle. We also have our mitochondria over here, responsible for generating lots of ATP. We have our lysosome here, which can digest unwanted substances. And then we have our smooth ER over here, which is going to have something to do with lipid synthesis. And then we have our peroxisomes, which have a number of functions, right? We can do chemical digestion. We can also do lipid synthesis. Ribosomes come in 2 different types. You have bound ribosomes. They're attached to the surface of the endoplasmic reticulum, right? So that's why we call it rough ER. But you also have free ribosomes. They're just suspended in the cytosol, and you can have proteins synthesized out in the cytosol as well. This is where the majority of proteins are synthesized, but you do have other options. Centrosomes. These are cylindrical centrioles surrounded by a protein, and these are going to function in cellular division that we're going to talk about in just a second. Cells also have proteasomes, and these are going to help digest proteins. So if a protein is damaged, if it's incorrectly folded, again, we're kind of taking out the trash, right? So the protein will be tagged for disposal, and we'll remove the protein through proteasome action. Another non-membrane-bound organelle is the cytoskeleton. This provides support, right? It organizes the organelles. It's also going to assist in cell division to help pull the cells apart. And then these are some of the filaments that would be included under cytoskeleton. So cytoskeleton is kind of the big umbrella, and then you have different classes of cytoskeleton, and they're going to have different jobs. Here we have some of the examples, and this figure does a nice job of kind of breaking it down for you, what each is involved in. What I usually think of is microfilaments. These are small. They're going to have something to do with cell division and helping to maintain the shape of the cell. Then we have these medium-sized intermediate filaments, and these mostly have something to do with cell communication. And then we have these large microtubules. These are going to be really important. Oops, I was supposed to write division. Really important in cell division, and they also help with movement. They help move items inside of a cell. I know we've talked about these structures already, the cilia, which are hair-like, the flagella, which is tail-like, and the microvilli, which are finger-like. But you're going to see on the next page that microvilli, these extensions happen because of microfilaments. You can see in this image here the microfilaments are what is actually driving the projection of the plasma membrane. Remember, microvilli are just loops in the plasma membrane. So how are those loops created? Well, basically we have microfilaments that are pushing in a specific direction on the plasma membrane and creating these extensions. Finally, we get to talk about these membrane junctions. So I'm talking about if you have two cells, how are these cells staying connected? And if you say who cares, remember we take cells and what do we make with cells? We make tissue. And then what do we make with tissue? We make organs. And what do we do with organs? We have organ systems, right? So you have lots of different cell types that are making up the tissue which help the organ function. So it depends on how we link the cells together. Do we want a tight junction where nothing can pass between them? Do we want a gap junction where we provide a direct passageway for substances to travel? Or do we want desmosomes, which are more like anchors? Here's the figure from your textbook, and it gives you an example of each of these junctions. Here we have the tight junction, right? The tight junction makes it impenetrable. Probably think about your stomach or your bladder, right? What do we do with the urinary bladder? We fill it with urine. Do we want the urine to be able to seep out of the bladder? No, we want it to go bladder and then ureter or ureter, and then we want it to exit the body via the urethra, right? So your bladder is a storage tank. We don't want urine to be able to leak out into your pelvic cavity. For desmosomes, these are more anchoring junctions. So if I want to have two cells stay close to each other to create tissue, but I still want to have some flexibility, you'll have these desmosomes or anchoring junctions. So skin is a great example of where you would see desmosomes. If you've ever gotten a sunburn and you've been able to peel off a sheet, right, of skin or tissue on the top epidermis, that is because the desmosomes are holding the individual cells together. Gap junctions. Gap junctions, there's a communication channel. Ions can pass through gap junctions. When I think of gap junctions, I usually think of the heart. I think the heart has to contract all at one time, or specifically the ventricles of the heart, to push the blood up and out of the heart and into your cardiovascular system. So there has to be a coordinated effort to contract the ventricles at the same time. So you want to allow the cardiac cells to talk to each other, and they talk to each other via ions. So you have to have gap junctions in cardiac muscle. Let's talk about the structure of the nucleus, and then we'll move into talking about cell division. So we've already described the nucleus, and we said inside it has DNA. Here they use the word chromatin. It's the same thing. It's both a series of letters of A, T, C, and G. Chromatin is just specific in that it's been wound really, really tightly so that we can pack all the information inside of the nucleus. So chromatin DNA, same thing, just has to do with shape. We know that the nucleus has this membrane. It has an inner and an outer membrane. We call it the nuclear envelope. So remember, we want to keep our DNA safe. Our DNA is behind a double door. We don't want anything to happen to the DNA because then that can affect protein production, right? This is your template for all your proteins. So the nuclear envelope has pores in it. Remember, we do have to take the DNA and we have to turn it into RNA. We have to copy it. Well, we want the copy to get out to the rough endoplasmic reticulum, because what does the rough endoplasmic reticulum do? It takes the RNA and turns it into an amino acid sequence or a protein. So your DNA is copied into RNA, and then your RNA is going to be threaded through a pore. And remember, right out here is going to be your rough endoplasmic reticulum. So we start the protein process. The other structure here that we haven't talked about yet is the nucleolus. I kind of hit it with all my zigzags. But here's the nucleolus. The nucleolus is responsible for generating the ribosomes that are going to help this process occur. So the ribosomes necessary to turn RNA into a protein come from the nucleolus at the core or the center of the nucleus. I skipped over some of the wording so that we could come straight to this figure because I think it makes a little bit more sense. So here's our typical DNA alpha helix, right? We've seen this structure before when we talked about the macromolecules of living things. And we see our A, T, C's, and G's here, our purines and pyrimidines attached to a sugar and a phosphate group. That was our nucleotide, right? So we see that the DNA gets wrapped around these things called histones, and we call this complex a nucleosome. So we have a bunch of DNA that we're wrapping around this histone and we're coiling it, and this is where that term chromatin comes in. And then chromatin is wound into a specific structure which we call a chromosome. Most of your DNA is going to stay in this chromatin state until you're ready to divide, and then we're going to organize and form this chromosome because it's easier to duplicate and split. So you'll see this coming up in cell division, but think of chromatin as kind of this ball of yarn. It'd be really hard to cut it in half and get equal parts. Where if we organize the chromatin into chromosomes, I can split it right down the middle, and this arm can go this way and this arm can go that way. So we know that DNA copies into RNA, and then RNA is processed into proteins or an amino acid sequence. It's a really complex process. They have official names, transcription and translation. So transcription is taking DNA and copying it to RNA. That's transcription. Translation is taking that RNA and turning it into a protein amino acid sequence. And there's a lot that they've kind of skipped over here. I just want you guys to know the basics, be able to tell me that DNA is transcribed or undergoes transcription and turned into RNA. I would also think about where you would find these. So DNA is always in the nucleus. If it leaves the nucleus, it can be broken down and degraded. We don't want that. That's our template, right? The RNA can be found inside the nucleus because we had to copy it from DNA. And then we thread the RNA out of the nuclear pore. So the RNA can also be found out here in the cytosol or the endoplasmic reticulum where it's being translated into a protein sequence. Now we're going to talk about cell division. So if I have one cell, and it needs to copy itself. I need to make another copy. That's cell division. Think if you cut your skin, right? You accidentally walked into a tree branch and you left a gash on your arm. Well, we have to fill in that gash with skin cells. So what do? We take the existing skin cell and we copy it and we fill in where the missing skin cells are located. So this would be an example of mitosis, cell division that occurs in somatic cells. Somatic to me means body. So in body cells, except for sex cells. So what happens in sex cells, or what are sex cells? These are the cells of the ovary and the testes. Ovaries are going to create eggs, and testes are going to create sperm. So mitosis is happening in all of your body cells. When we go to make egg and sperm, we have to do things a little differently. We have to do meiosis. I think I briefly mentioned this in chapter 1. We talked about mitosis and meiosis. So all body cells are doing mitosis. What about meiosis? What are the only cells or the organs, the cells within them that are doing meiosis? So you only find meiosis happening in the ovaries and the testes. The cell cycle is the process, the name of the process that we use to describe the replication of body cells. So basically, if you have one cell and it needs to copy itself, we're doing the cell cycle. So there's interphase, which lasts the longest. It's kind of the prep phase, is what I call it. I'm basically prepping to divide. And then the mitotic phase is the actual division. So lots of prep to do a quick little division. Here's an image of the cell cycle from your textbook. Everything in blue here, this is all interphase. This is all of the prep. And then here is mitosis, or the meiotic phase. That's the actual division. So what's happening in these phases? First, we have G1. This is where we're growing the cell in size, and we start replicating the organelles. Remember, if you have a cell that's got 5 mitochondria, if you need to create another cell that has 5 mitochondria, you have to copy those, right? If not, you're just splitting everything in half. So this is where we have the growth phase. This is where we're getting everything ready to replicate, copy the DNA, and we're creating the organelles, right? S phase is where we actually copy the DNA. So we take DNA and we copy it so that we have 2 copies of all of the DNA. And then G2 phase is where we're going to continue to grow and we get ready. We put everything in place for division. Then in mitosis is where we actually have the phases, and they're listed here: prophase, metaphase, anaphase, telophase. And then cytokinesis happens where the cell actually splits, and now you have 2 daughter cells. And daughter cells doesn't reflect sex, it's just what we call them. These are the first cell, this is the parent cell, right? And so then these are the daughter cells. Now we lead into cell aging. So we have our cell cycle that we just looked at, and we have our G1, our S, our G2, and then our mitosis phase or meiotic phase. So the cells will go through the cell cycle repeatedly, but after a period of time, as you age, you're going to have a lower number of functioning cells. You're going to have some more abnormal cells that just didn't happen to get digested by lysosomes, or they were never tagged as abnormal. You can have different numbers of organelles. So when people say, oh, the older I got, my metabolism started declining, that could be due to hormones, right? That could be due to metabolism that's happening inside of the cell, which could be due to the damage of some of the organelles. You're also going to have structural changes in your DNA, right? The more we're exposed to environmental carcinogens or things that cause cancer or that damage our DNA, the less likely we're going to have healthy cells that function normally. Even though cells are aging, we do have a way to kind of stop cells from propagating or copying themselves if they're damaged. So we have this process called apoptosis, which is programmed cell death. So this occurs normally, this happens throughout your lifetime, where you destroy or remove cellular components and cells if they're defective. So if you have anything like DNA damage, we don't want the cell to copy itself if its DNA is damaged, right? If you have an organelle that's not working correctly, you can trigger apoptosis. Remember we talked about lysosomes? Well, the lysosomes can open up and release their contents, their digestive enzymes, and basically it's an auto destruct sequence for the cell. Apoptosis can destroy the DNA polymerase, which copies DNA. It can actually digest sections of DNA. It can digest the cytoskeleton. It can shrink the cytosol and destroy organelles. So I want you to realize that apoptosis is a good thing if we want to clear cellular debris, and it's important during proper development. You have to have apoptosis to remove some extra unwanted tissue when you're developing limbs. It can also remove cells that are harmful to you, right? They're not functioning properly. They're just taking up space and producing lots of maybe toxic waste because they're not working properly. So apoptosis is a good thing. It is programmed cell death to kind of get rid of unwanted substances. But as you age, we can't just keep doing apoptosis. We'd get rid of all of our cells that don't function, right? So basically, as you age, all of your cells may not work as effectively, and some of these processes just kind of continue on this decline throughout life. Yay! Happy ending to chapter 4, right? All right, I'll talk to you guys later. Bye.